Good morning, everyone. My name is Carolyn Hogg and I'm from the University of Sydney, and I'm here today to talk about our regional update for Australian Biodiversity Genomics. And the work that I'll be presenting today is by no means my own. It is really a accumulation of what Bioplatforms Australia, the Australian Biocommons and the Atlas of Living Australia are contributing to Australian biodiversity genomics, as well as a range of other academic and other conservation scientists that work in our region. So I hope I managed to do everyone justice here this morning. So Australia has pretty unique biodiversity. We have about 8,000 different vertebrate species found in Australia, in tens of thousands number of invertebrate species, as well as approximately about 24,000 different plant species. But the amazing thing about Australia is that we also have a large percentage of endemic species. So these are species that are obviously found nowhere else in the world. And so really the ability to create genomic resources for these species, as well as generate genomic data and help conserve our biodiversity in the long term, is really the responsibility of those that reside within our nation. So Bioplatforms Australia is a nationally funded, government-funded agency that is about promoting and developing omics infrastructure here in Australia, whether that be metabolomics, proteomics, or genomics. And part of what they do has an environmental or a biodiversity theme. And today, what I wanted to do is just run through some of these different biodiversity initiatives that Bioplatforms has been undertaking for about the last 10 years and show you the depth and breadth of some of the work that is occurring here in Australia. So one of the first biodiversity initiatives that Bioplatforms funded was a co-initiative with the Great Barrier Reef Foundation that ran from about 2012 to 2016, and they have generated 9 reference genomes for some coral species as well as some symphodiums. And in 2018, there was an investment, and a few years before that, both the koala reference genome was published as well as the cane toad genome. So although the cane toad is not a native species to Australia, it is an invasive pest species that has caused significant damage to Australian biodiversity from a range of different frog species have been lost, as well as reptile species and birds that consume the toxic cane toads. And so we really needed to have an understanding of the species to help inform management actions. On the back of the koala reference genome project, Craig Moritz and Rebecca Johnson took on the AusMammal Genomics project, which ran from about 2016 to 2021. And during the course of that project, they've developed 15 reference genomes for a suite of different marsupials, as well as rodents and other mammals, including bats here in Australia. And one of the key aspects of this project was developing reference genomes, but was also developing an exon capture, which allow us to do taxonomic resolution, not only for the rodents across Australia, but also a large number of marsupial species. And that work is just being finalised at the moment. And also AusMammal Genomics invested about 1/3 of their money into population genomics. On the back of understanding what's happening with our animal and vertebrate species, there's also been an investment into the genomics for Australian plants, as well as the Australian grasslands. There's a new project that just commenced in 2020. There's been about 10 reference genomes published for plants. As you all know, for those of you who work in plants, plants are very complicated to do when it comes to genome assembly, particularly around the polyploidy issues. And again, like AusMammal Genomics project, there was a number of projects in gap around taxonomic resolution, as well as some population genomics. This, of course, then leads me to 2 of the big biodiversity initiatives that have been going on that started during the pandemic. In 2020, Craig Moritz and Arthur George, as you can see pictured here, started the AusArg, or the Australian and Amphibian and Reptile Genomics Project. To date, they've sequenced about 17 reference genomes, and they have about another 5 reference genomes being externally funded, not by Bioplatforms money, but are linked back to the AusArg project. One of the critical things for the AusArg project is actually taxonomic resolution. There's a lot of uncertainty around taxonomy in Australian amphibians and reptiles, and Craig and Arthur and the teams involved in AusArg are working very hard to try and resolve that taxonomic resolution, and also investing in some population genomics for some of the species that they were working on. For those of you who may not be aware, I head up the Threatened Species Initiative, which we also started in 2020 at the same time as Craig and Arthur with AusArg. We've done about 21 reference genomes to date in our pilot and our phase 1 part of our project, as well as population genomics. We're still working really hard to develop the Applied Conservation Genomics Hub. There's been some glitches with trying to make an online portal which makes it easier for conservation managers to access the data sets. And we think that there'll be about another 12 reference genomes for threatened species coming along in phase 2. And the Threatened Species Initiative is very much geared around developing genomic resources for threatened species. So it's not just about reference genomes. Some of the time it's just about producing genomic resources, whether it be reduced representation sequencing or whole genome resequencing. We've got about 62 species in the project, in 40 projects with about 120 collaborators to date. We're just about to announce the phase 2 round of funding, which will be another suite of species. And as you can see, they're representative of the different IUCN threat categories, from extinct in the wild through critically endangered, endangered and vulnerable. And we've also invested in some data deficient species where we simply just don't have enough information to be able to determine their threat status. As representative of Australia's biodiversity, we have a large proportion of species that are in fact plants, and then also across a range of taxonomic groups. And pleasingly, about a third of the project is only academics, and then the rest are government and conservation agencies. So this has really shown the need and the uptake of the conservation managers about biodiversity protection in Australia and how they can use genomic resources to further inform their decision-making and policy around biodiversity protection. There's also a number of biodiversity initiatives are more at the environmental DNA level. Bioplatforms of Australia has just started investing in the National Biodiversity DNA Library Initiative. This is being led by Oliver Berry at CSIRO, as well as an understanding of the biome of Australian soil environments and plant pathogens, which have a significant impact not only on our native species, but also on our agriculture. So one of the other steps we've been taking is trying to consolidate where the data is produced and genomes being produced in Australia. And we've set up the Australasian Genomes data portal with the AWS Open Datasets program. So this is very similar to where the Vertebrate Genome Project are launching their genomes. And over time, our aim is to start to try and pull more of the Australian genomes together in one location that are being produced by bioplatforms initiatives. There are a range of other biodiversity initiatives that are currently being undertaken in Australia. Arthur Georges and Andrea Killian are working together with Diversity Arrays. Andrea is from Diversity Arrays and Arthur is a strong collaborator of theirs, developing the One Dart project, which is about how to utilise the population genetic data that Diversity Arrays is generating and making it more accessible and available to not only the research community, but the conservation end users. Purwinder Kerr from the University of Western Australia heads up DNA Zoo Australia, who's been generating short-read high-C genomes for a suite of different Australian species and making those publicly available for others to then build on when they're trying to make informed conservation decisions. Chris Austin from Deakin University has a genomics hub down there, and they've been doing low coverage genomes, particularly for a suite of different crustacean and other arthropods in Australia. And Owen Edwards from CSIRO is the representative for Revive and Restore here in Australia. So it's not just the bioplatforms initiatives, but there are a suite of other initiatives that are occurring in Australia about biodiversity genomics and protection. One of the things we've come to realise recently, however, is there is quite a large number of genomic resources available across the world for Australian species, but it's actually very difficult to correlate this information and easily access it. And so Bioplatforms Australia, myself, the Atlas of Living Australia, the Australian Biocommons received funding from the Australian Research Data Commons this year to build what we're calling ARGA, or the Australian Reference Genome Atlas. And ARGA is really being built on the back of the Atlas of Living Australia infrastructure, which is essentially a way of using API technology to source information from across the world and then correlate it in one place. So you can go into ARGA, you can type in your species and ask to search for all mammal species, or whether they've got any functional traits or their threat status, their habitat type. It's a range of different options. And it'll pull up your species. You can see the koala here. give you the species distribution records, where they sit in the phylogenetic tree, if that has actually been resolved. So this is going to be built on the back of some of the other phylolink work within the Living Atlas, which is also building on some of the taxonomic work that's being done through the bioplatforms initiatives. You'll be able to locate where the genome assemblies are, where the raw data is, and any other omics data that might be available. So you can then go into the genome assembly. You can either download it if it's located on NCBI or wherever it's located. You can open it in a genome browser, or you can actually download the raw sequences yourself if that's the type of information and assembly that you're wanting to do. And one of the critical things we've been trying to do is make it more seamless and easier for people to move large data sets around Australia, particularly when people work at non-academic organisations such as museums or within government agencies, which makes it exceedingly difficult if you don't have access to large compute resources. So one of the other things we've been investing in pretty heavily in Australia in the last couple of years is how do we develop compute resources, not just for the academic sector, but for anyone who's interested in working in biodiversity genomics. So because many of the initiatives and many of the reference genomes and other genomic resources that are being developed in Australia are actually being developed through partnerships with bioplatforms and the biodiversity initiatives. A lot of the raw data actually sits within Bioplatforms Australia. So this is where we decided to start. And working with the Australian Biocommons and the National Compute Infrastructure, which is the National Computing Infrastructure in Canberra, NCI, Galaxy Australia is located through Melbourne University down in Melbourne, and Pawsey Supercomputing Centre is over in Western Australia. And what it's allowed us to do is we can now actually take data directly from bioplatforms straight into Galaxy Australia to allow us to undertake the Hi-Fi ASM PacBio genome assembly. NCI has been working pretty closely with the AusArc team on how to do Oxford nanopore genome assemblies. And we've also been working on a population genomics pipeline with Galaxy Australia. We only have a couple of licenses for genome annotation, but that all sits with Pawsey Supercomputing. And so what we've been trying to do is develop pipelines and tools with associated documentation. This is all pulled together by the Australian Biocommons team, which is allowing other researchers who are new to the genomic space on how to take their data, easily access it, and build their genome assembly. As many of you know, our team has been working with Amazon Web Services and Ronin. We've been trying to work out ways to do efficient genome assembly, particularly of large genomes like amphibians in the cloud, as well as our transcriptome assemblies. And this is where the TSI conservation Applied Conservation Data Portal will be within the AWS cloud space, because that again allows anyone who doesn't have access to large compute technology to be able to run the analysis pipelines. And finally, of course, we have ARGA, which is our new reference genome atlas, which has just started its current build. That's going to go for the rest of 2022 and all of 2023. And of course, as well, we have Australasian Genomes, which is the AWS Open Datasets program. So I guess this is a lot of information in a very short span of time, but so where are we at with Australian biodiversity genomics? So if you're an end user standing there scratching your head, once we've built the Australian Reference Genome Atlas, life will become much easier. And the reason for that is you'll be able to go there, type in your species and find out what information is available, if any, for that species, and that'll be drawing on both the national and international data repositories around genome assemblies. You'll at least know whether or not you have a reference genome for your species, and we'll also start drawing on the bioplatforms data portal and hopefully the OneDart portal once they've completed that work as well. And so to actually know where population genomic data exists as well. If, however, you go to the Reference Genome Atlas and your species doesn't in fact have any data, you can then work with the bioplatforms initiatives to generate your data sets, the National Compute Services to assemble and annotate the genomes, and then you can put the information into, with your population genomic data into the TSI portal and be able to get your standardized conservation reporting. So this is the dream. The bigger dream is to actually, if the data is already in existence, it can be correlated together and you can pull it into the portal to run your analysis. So this part of the project will keep going in all 2023 and 2024. We're working with the Australian Biocommons team and the Australian Academic Federation at the moment to work if we can have a way where we have one login, which allows you to go into Australian Biodiversity Genomics and you can access the data portals, be able to access all the compute resources you require, as well as the TSI portal, and kind of make it a one-stop shop, but that's still living the dream. So this project is by no means just me. There is a large number of people involved in this project, from the end users, those who provide the samples, the compute and the sequencing technology. And much of the work is funded by the government agencies as well as the Australian Research Council. So thank you very much, and feel free to ask any questions. Just send me an email. Thanks a lot. Bye.